For this last video clip for Chapter 8, I'm going to talk a little bit about genetic engineering, something we hear about all the time in the news. If not genetic engineering, you certainly have heard of GMOs, or genetically modified organisms, and we mentioned that a little bit back in Chapter 1. And so I'm going to just kind of hit some highlights on some of the topics from this part of Chapter 8, some things that I want you to be familiar with, and as with some other parts of this chapter, you are going to read and be exposed to more of this information as you're going through the chapter, but use your study guide, learning objectives as guidelines for what you need to know for testing purposes. In a single semester microbiology class, we just can't cover everything in depth. But biotechnology is something that's a very hot area for the 21st century. And what is biotechnology? Biotechnology is using biology in a technology type way to try to improve the quality of life on our planet. So that could be from coming up with new types of medical treatment standpoints. It can be improving the environment, improving foods and nutrition, and so forth and so on. So biotechnology has a lot of applications. So development of new therapeutics is one area of biotechnology that's really, really important. And I alluded to that at the beginning of this chapter. I was telling you, you really need to kind of take this chapter seriously, because medicine will be transitioning to more of a genetic type approach during the careers that you all have in healthcare. So you need to have a little bit of foundation in this area as you move forward. Development of new therapeutics. So that's going to involve things like, eventually we're going to be able to correct genetic disorders. If you know somebody who has a genetic disorder like cystic fibrosis or muscular dystrophy, or sickle cell anemia, we mentioned that in the mutations video lecture. Those are things that ultimately down the road, they're caused by mutations on the DNA. Hopefully down the road we'll have the technology to correct those mutations. We're making progress in those areas, so that will involve new types of therapeutics that not too long ago seemed like something out of science fiction. And there are people who are looking at things. There are companies here in Huntsville who are looking at things like engineering viruses, so they will go in and attack and kill cancer cells. So medicine is going to be changing quite a bit. Diagnostics, so figuring out what is causing a problem in a patient. A lot of those types of things are moving more toward genetic testing. There's a company here in Huntsville called Diatherix, and their claim to fame is they have come up with these genetic-based tests that allow very, very specific identification of bacteria and viruses that are causing infections in patients. So that's another new area that's changing a lot. Anytime we use microorganisms or viruses for our benefit, that is a type of biotechnology. Even things as simple as making yogurt, making beer and wine, those are biotechnology applications. From a more modern standpoint, we are able to genetically engineer or modify crops so that they have improved drought resistance or pest resistance, or you can engineer foods like rice so that they produce vitamins that they did not produce before. And so that all involves biotechnology. That gets into genetically modified organisms, which some people have an immediate negative reaction to, but you can't really throw the baby out with the bathwater. You have to weigh the pros and the cons and really look at how is that genetic modification benefiting humankind. A lot of times the pros may outweigh the cons of doing that. So keep an open mind about that subject. There's kind of a throw the baby out with the bathwater mentality out there about it. Making drugs, hormones. If you know somebody who takes insulin because they're a diabetic, insulin is produced by Escherichia coli bacteria that have been genetically modified to make human insulin. So that's another area of biotechnology. Many things that we take on a daily basis are biotechnology products. And I just mentioned replacing defective genes. We can't do that all that well yet, but hopefully not too far down the road we'll have these technologies that allow that to take place. And everybody, on another note, everybody's interested in helping humankind break our oil habit, not just from the standpoint of reducing fuel costs, but also helping to protect the environment. And so, will microorganisms give us new ways to fuel and power our transportation and our other, and help supply other energy needs that we have. You guys have no doubt heard of biodiesel. That's where plants of some type, crops of some type, like corn or grasses, those are taken, and then bacteria ferment the carbohydrates in that plant material and generate ethanol. That's your biodiesel. So that's a new method for fuel production. And then we have microbial fuel cells. This is kind of a little bit more space-agey, where it may be possible down the road microorganisms are involved in energy generation processes, just like our cells are, and we've talked about that back in chapters 6 and 7. And can we harness that ability in some way? Can we have microbes growing inside a little vat and converting the energy, the chemical energy stored in carbohydrates that they're breaking down through catabolism, and converting that to other forms that could be used to do things like power your car? So who knows, maybe somewhere down the road, instead of filling your car up with gas, you'll stick a microbial fuel cell into the engine, and these microbes will generate the power for you. Another area of biotechnology is called bioremediation. And here it's, remediation, that term, like if you had to take a remedial class, you're trying to improve your basic skills. So here bioremediation is referring to improving, cleaning up the environment. We've had a great example of bioremediation within the last few years. Along the Gulf Coast, you had the big Deepwater Horizon oil spill. Blanking out on the name there for a minute. The Gulf oil spill a few years ago. All this oil got dumped out into the Gulf of Mexico, and of course it was a huge cleanup nightmare. However, a lot of the oil was digested much more quickly than anybody anticipated, and it's because there are bacteria that naturally live in the Gulf of Mexico that are capable of catabolizing the hydrocarbon molecules that are present in oil. They can actually use that as a source of nutrition. Would be toxic for us, but it's food for them. And so all these, when all that oil got spilled into the Gulf of Mexico, all those bacteria suddenly exploded in population. It was like they were at a breakfast buffet, and it helped clean up the oil spill. Now down the road, we can research that, and maybe we can deliberately use those microorganisms to help clean up future oil spills. And there are microorganisms that are being looked at to help clean up other types of toxic chemical spills and other types of environmental problems as well, because there's so much diversity out there in terms of what microorganisms can use as their energy sources. What can they break down? So bioremediation gets into applying that information to help us solve environmental problems. Okay, your textbook has a section in the book where it talks about gene cloning. And now this is not cloning like we're cloning a whole human being, and I'm going to have like a little mini-me version of myself through cloning. This is gene cloning, so you're cloning an individual gene that codes for a protein, and you're moving that from one organism to another. That's an aspect of biotechnology as well. So if you engineered rice to produce a vitamin that it could not produce before because you took a gene out of bacteria and put that into a rice plant, that would be a type of gene cloning. You have cloned that gene from the bacteria that can produce a vitamin, and you have inserted it into a plant. And that's also an aspect of recombination, because we are recombining genetic information in new ways. And we discussed how that can occur naturally through horizontal gene transfer, but it can also occur artificially in the laboratory through biotechnology type purposes. And this is a table from your textbook, which, and take a look at this. I'm not going to go through all of the details on it, but take a look at this in your textbook, and you'll see how develop a little familiarity with how a gene that codes for a particular protein can be taken out of something like a human cell. Like this could be a gene for production of insulin. Insulin is a protein hormone that's very critical for control of blood glucose levels. And then what you can do in a laboratory in the test tube is take that gene and insert it into a plasmid, one of those little extra pieces of circular DNA that can grow in some types of bacteria. And there are techniques for doing that. There are enzymes that you can use that are like cutting and pasting type enzymes to help you do this. It's like almost like working with little, you can't see what's going on because these are molecules that are invisible to the naked eye, but you're using these enzymes that are kind of like little molecular scissors and molecular pasters, and that's allowing you to put pieces of DNA together in new ways that didn't exist before. Then once you have this little plasmid, this little circular piece of DNA with this gene from a human, this gene that codes for insulin, you can artificially insert that into bacteria. Escherichia coli is the most commonly used species for these types of things. And now the bacteria will express that gene. So you go through transcription and translation, and now you're making your human insulin or whatever protein product it is. And so you can do that on a very large scale. You can grow up big vats of these bacteria that crank out large amounts of these protein products, whatever they may be, and you purify those protein products, and then you use them. So your textbook will talk a little bit about restriction enzymes. These are naturally occurring bacterial enzymes that we have been able to harness because you can use them like molecular scissors that cut DNA at very specific locations to help you with all this cutting and pasting that goes on as part of gene cloning. When you hear about genetically modified organisms, again, those are organisms that have received, through gene cloning techniques, a gene or genes from some other organism, and that's why they have been modified. All right, a biotechnology technique that I'm not going to hold you responsible for learning the gory details of how it works, but I want you to be familiar with it, is called PCR, and that's an abbreviation for polymerase chain reaction. And this is referring to DNA polymerase. Remember, that's the enzyme that builds DNA molecules. And a chain reaction, you think about a chain reaction, that's something that amplifies. So this is a very sensitive, a very powerful laboratory technique. It's being used more and more even in clinical laboratories, so you're probably going to hear about patients who have submitted a sample and there's a PCR type test being done on it to try to detect something or diagnose something. And it's a very specific technique, and what it does is it targets a specific stretch of DNA, like a gene from a virus, and finds that DNA that might be present in a sample in very minute quantities, so low that you can't detect it. But then this polymerase chain reaction technique will artificially amplify that target DNA. It'll make many, 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 many copies, billions and billions and billions and billions of copies. So you're amplifying this DNA artificially in a test tube, and now you've got enough of it to actually detect it, use techniques to actually see that it's there. And one of your lab activities, you're going to do an activity where you do DNA agarose gel electrophoresis and separate DNA on an agarose gel. So when you do that activity, think about PCR, because when I'm telling you can actually detect DNA, that's a common method used in the laboratory for actually being able to visualize DNA molecules. So there's lots of techniques for this. Clinically, it's being used a lot for diagnostics. So I just mentioned the company in Huntsville over in the Hudson Alpha Biotechnology Institute, and they have developed lots of PCR tests that home in on genes of particular viruses or bacteria that are present in a sample at very low levels, so there's no way to really clearly detect them. You use this technique and it very specifically amplifies, say, a gene from papillomavirus or a gene from Shigella bacteria, and amplifies it so much that you now have enough to actually see it through some sort of laboratory technique. And that can kind of give you a yes-no answer. Yes, this patient had this virus present in the blood. This patient had this type of bacteria present in a fecal sample, so that's what's likely causing infection. So the use of this technique in diagnostics is going to be huge. All right, now that was just kind of a very general summary. Take a look at this table from your textbook, which is trying to give you a visual on how PCR works. However, the best thing to do is to go to this link right here, PCR Virtual Lab, so you'll need to click on that. In the full version of the PowerPoint that's available on Blackboard, and if you click through that, there's an animation there that takes you through the PCR process step by step, and when you see it animated, it makes a lot more sense. Just kind of in a nutshell what happens. Here you've got a DNA sample, and during PCR there are three major phases. One is denaturation, which means the DNA double helix unwinds, it separates. And you have designed primers, PCR primers. These are little stretches of A's and G's and C's and T's that specifically match up with a particular area of DNA that you want to amplify. You design those. You can order them from companies. There's a company right here in Huntsville called Eurofins MWG Operon that this is one of their claims to fame, is they make these little pieces of single-stranded DNA that you go on the internet and type in what you want them to be, and they'll send them to you by FedEx about 24 hours later, and they're not all that expensive. And you use that for this particular technique. So all this is happening in a little tiny test tube, and those primers that you've designed, through complementary base pairing, they match up with an area of DNA, like that could be DNA from some virus that you're interested in detecting, like let's say herpes virus. That's called the priming step. And then there's an extension step where you have DNA polymerase in this test tube, and it latches on to the ends of the primers, there's your polymerase, and rides along, and through complementary base pairing, it synthesizes new DNA strands. Then you go through each of these cycles again, over and over and over. So you can start off up here with a very low amount of DNA, but through this denaturation, priming, and extension series of steps that takes place in the test tube, that those relatively few copies can pretty quickly build up and become hundreds and then thousands and millions and billions and even more copies of the DNA. And then, as I mentioned, then you've got enough that you can, there are techniques that you can use in the laboratory, like agarose gel electrophoresis, to actually visualize the DNA. So kind of the key things, though, in terms of what you need to know about PCR for testing purposes, use the review guidelines and the study guide to help you home in on that. And take a look at this PCR virtual lab online, because it's got an animated explanation is going to work a whole lot better for you. Now, I actually mentioned this is another area of biotechnology I want you guys to be familiar with, because you may be hearing about this not too far down the road in clinical laboratories. And I mentioned this earlier when we went over the different types of RNA. Ribotyping is a new way of identifying organisms through DNA sequencing techniques. So DNA sequencing has become very powerful. We can pretty quickly determine the DNA sequence of the entire genome of a bacterial organism very quickly. Any type of organism reasonably quickly, but bacteria tend to have pretty small genomes, so their entire genomes can be sequenced very quickly. I mentioned the blue structures here used in translation. Remember those? Those are the ribosomes. And we talked about how a ribosome can be broken into a large subunit and a small one. And the small subunit in bacteria has a piece of RNA called the 16S ribosomal RNA. That's part of one of the building blocks of that ribosome. There's some proteins in there as well. That RNA molecule is going to be about 1,540 nucleotides in length. And I mentioned before that those 1,540 nucleotides, that sequence will vary from species to species. So today it's not too difficult to determine. If you have a sample of bacteria from a patient, you can pretty quickly determine the sequence of the 16S ribosomal RNA from that organism, and then you compare it to a computer database, and out pops the identity of your organism. And so eventually we're going to get to the point where that is less expensive and much faster than the types of techniques that you guys got some experience with in your virtual lab on unknown bacteria identification. So something like this will ultimately substitute for all of those biochemical tests and that have traditionally been used to help us identify microorganisms in the laboratory. And all living things have ribosomal RNA, so it's not 16S in prokaryotes, it's going to be 18S, but there are similar types of techniques where you could use this technology to identify fungal species or helminths or protozoans or whatever. And it really is a type of DNA sequencing. I know we're saying, well, 16S ribosomal RNA, that's what we're determining the sequence of. But these RNAs also have genes on DNA. So these RNAs are made through transcription. There's a ribosomal RNA molecule there. So really the sequencing is being done on the DNA version of the gene that codes for your 16S ribosomal RNA in bacteria. So just be familiar with that as well. Ribotyping is a method that's ultimately going to be used to help us identify organisms that are causing problems for patients and other applications as well. All right, so we're finally finished with Chapter 8. Next, we're going to backtrack. Now that you have some background in genetics and you're a genetics expert, we're going to backtrack over to Chapter 5 and talk more about viruses. I skipped over Chapter 5 because I really feel you got to have some of this background in microbial genetics so that the things we discuss in Chapter 5 will make more sense for you. So the next video clip will be moving backwards to Chapter 5.